Okay, next we are going to study how selectable marker help us to identify transformants from non-transformants and recombinants from non-recombinants. Okay, so there are two types of selectable markers. One is, as in the case of PBR322, antibiotic resistant gene acting as selectable marker. And in E. coli bacteria, there is a natural selectable marker, that is a chromogenic selectable marker. Means it is identifying the recombinants by the absence of color. Okay, so first we will study how antibiotic resistance gene in PBR322 help us in identifying transformants and recombinants. Okay, so ഇത് നല്ലവണ്ണം ശ്രദ്ധിച്ചു കേട്ടില്ലെങ്കിൽ you will get confused. ബുദ്ധിയുള്ളവർക്ക് മാത്രമേ ഇത് മനസ്സിലാകത്തുള്ളൂ. So let me see how many of you are intelligent enough to understand this. So our aim is to make a recombinant E. coli bacteria. That is, first of all, we will take PBR322, which is an artificial E. coli vector, synthetic vector, to which we are adding the foreign gene. Okay, that is the gene which we are in need of, or a desired gene is added to the PBR322. So when we add this desired gene to PBR322, or PBR322 may not take up this desired gene. So some of the PBR322 will be without any desired gene as it is. Means how it is having 4 genes, that is ampicillin resistant gene, tetracycline resistant gene, ROP gene, ORI and ROP gene. All the 4 genes will be functional. അങ്ങനെയുള്ള കുറേ E. coli bacteria കാണും. That is E. coli bacteria which have taken up the plasmid PBR322, but it has taken up the plasmid without a foreign gene insert. We are going to insert the foreign gene in this tetracycline resistant gene. അപ്പോൾ ഇത്രയാണ് tetracycline resistant gene. അതിൽ BAM-H1 ന്റെ സൈറ്റിൽ, that is ആ BAM-H1 എന്ന് പറയുന്ന റെസ്ട്രിക്ഷൻ എൻസൈമിന്റെ റെക്കഗ്നിഷൻ സൈറ്റിൽ നമ്മളൊരു കട്ട് ഉണ്ടാക്കി. അവിടെ we have added a foreign gene here. So we had added a foreign gene in PBR322 like this. അപ്പോൾ നമ്മൾ ഒരു കുറേ PBR322 എടുത്തിട്ട് അതിൽ foreign gene add ചെയ്യുമ്പോൾ all PBR322 will not take up the foreign gene. A few of them will remain as such without any change. Some others will take up the foreign gene like this. Okay, so ഇങ്ങനെ foreign gene tetracycline resistant gene എടുത്തു കഴിഞ്ഞിട്ടുള്ള PBR322 will lose tetracycline resistance property. എന്ന് പറഞ്ഞാൽ എന്താണ് അർത്ഥം? ഈ PBR322 tetracycline resistant ജീന്റെ ഇടയ്ക്ക് ഒരു ഇൻസേർട്ട്, അതായത് ഫോറിൻ ജീൻ വന്നു കഴിയുമ്പോൾ ഈ ജീനിന് അതിന്റെ പ്രോപ്പർട്ടി നഷ്ടപ്പെടും. ബിക്കോസ് ഇൻ ബിറ്റ്വീൻ ദ ജീൻ എന്ന് പറഞ്ഞാൽ എന്താണ്? കുറെ സീക്വൻസ് ആണ് വിച്ച് ഹാസ് എ മീനിങ്. അതിന്റെ ഇടയ്ക്ക് വേറൊരു ഇൻസേർട്ട് വരുമ്പോൾ ആദ്യം ഉണ്ടായിരുന്ന ആ ജീൻ ഫംഗ്ഷൻലെസ് ആയിപ്പോൾ. അതിനാണ് ഇൻസേർഷണൽ ഇനാക്ടിവേഷൻ എന്ന് പറയുന്നത്. So now the tetracycline resistant gene will get insertional inactivation. So it will not function. But ampicillin resistant gene is functional. The ROP and other 2 genes are also functional. We need not mention about them because they are not the selectable markers. So these 2 are the selectable markers. So when the insert is there in the tetracycline resistant gene, this gene will become insertionally inactive. That is, it undergoes insertional inactivation and it will lose the tetracycline resistance property. Okay, so now we have to think that now we have plasmids, a few of them with the insert which has lost tetracycline resistance property, and few of them without the insert which has both ampicillin resistance and tetracycline resistance property. Now we are adding this mixture of plasmid of both type to the bacterial colony. ഈ കോളനിയുടെ ബാക്ടീരിയൽ കോളനിയിലേക്ക് നമ്മൾ ഈ പ്ലാസ്മിഡ് ആഡ് ചെയ്യാണ്. So some of the plasmids are with the foreign gene, others are without the foreign gene. നമ്മൾ E. coli ബാക്ടീരിയയിലോട്ട് ആഡ് ചെയ്യുമ്പോൾ some of the E. coli bacteria will take up the plasmid. Some others will not take up plasmid. Okay, ഇനി അപ്പൊ കുറെ എണ്ണത്തിൽ പ്ലാസ്മിഡേ കയറത്തില്ല, കുറെ എണ്ണത്തിൽ പ്ലാസ്മിഡ് കയറും. പക്ഷെ എടുത്തിട്ടുള്ള പ്ലാസ്മിഡ് ഏതാണ്? ഫോറിൻ ജീൻ ഇൻസേർട്ട് ഇല്ലാത്ത റീകോമ്പിനന്റ് വെക്ടർ അല്ലാത്ത പ്ലാസ്മിഡ് ആണ് ചിലതെടുത്തിരിക്കുന്നത്. ചിലതെടുത്തിരിക്കുന്നത് നമുക്ക് ആവശ്യമുള്ള റീകോമ്പിനന്റ് വെക്ടർ ആയിട്ടുള്ള പ്ലാസ്മിഡ് ആണ്. Okay, so 3 types are there. So, this medium, bacteria medium, plasmid add. Then we are going to identify. We need only the recombinants. We don't want the non-transformants and non-recombinant transformants. Now, in colony, white bacteria. They are without any PBR 322. In half black it is having plasmid PBR 322, but it has taken up the PBR 322 without any foreign gene. So now it has an additional quality that it has resistance to 2 antibiotics, that is ampicillin resistance and tetracycline resistance. Now the third group I have made it as dark, fully dark. These three are actually the needed ones. They have taken up the plasmid with the foreign gene. But what happened? Foreign gene or plasmid, it has lost the property of tetracycline resistance. That is, tetracycline and the antibiotic resist the property loss. But it can resist the antibiotic ampicillin. There are 3 types of bacteria immediately. Now we are going to identify one by one. Okay. Finally, we need only the recombinant. Now, we have 3 The white non-transformant because they have not acquired any foreign gene. Half white half black they are transformant but non-recombinants. Okay, non-recombinant transformants. Dark color the real recombinants. Now we will add ampicillin to this media. Now what will happen when we add ampicillin? It's an antibiotic. So when antibiotic is added to the media of bacteria, these white colored bacteria which are non-transformants, they cannot survive because they do not have ampicillin resistant gene. So what will happen? They will not survive, so they are removed. Now we have the media. So now what we have in the media are all transformants, means they are all having PBR 322. Some of them are having a recombinant plasmid, others are having non-recombinant plasmid inside. Now we want to identify the real recombinants. Now we will add tetracycline resistant gene. Actually, we are not adding, we are block plating in the technique. That is block printing dress or block color print chamber, blotting print chamber, print plating. Bacterial plating, bacteria grow bacterial plating, culturing. Now, this culture, we bacterial culture plate mediathil tetracycline adding. Tetracycline is another antibiotic. So we will be adding tetracycline to that medium. And then the same bacterial culture. Press real recombinants cannot grow. Why they cannot grow? They have lost the tetracycline resistance property. So only this bacteria which can grow in tetracycline resistant medium also will get transferred to that medium. Transferred out. So what we get back in this plate is the real recombinants because they cannot grow in the tetracycline resistance medium. So this is how we identify the real recombinants. But this process is a cumbersome process or a laborious process. Why it is considered a cumbersome process? Means a difficult and laborious process. The reason is that here to identify the recombinants we require 2 bacterial culturing. And incubation so it is also time consuming. That is why we consider it as a laborious cumbersome process. But there is another method, that is selecting the recombinants using chromogenic selectable marker. This is a one-step process. Or step we can identify the recombinants. So here what we use is, we use some substrate which can produce color. Okay, so this E. coli bacteria is having, we have studied about different genes. LAC is a gene, structural gene is a LAC is a gene. They produce the enzyme called as beta galactosidase. So we use a substrate which can be broken down into a product which produce color. Okay, so a substrate which can be acted upon by beta galactosidase enzyme and can be made into a color producing colony. ആക്കാൻ പറ്റുന്ന രീതിയിലുള്ള ഒരു chromogenic substance ആണ് നമ്മൾ ഇവിടെ യൂസ് ചെയ്യുന്നത്. അത്രയും ഡീറ്റെയിൽ നമുക്ക് വേണ്ട സിമ്പിൾ ആയിട്ട് പറഞ്ഞാൽ എന്ത് പറയും? Here what we do is we give insert or we are inserting the foreign gene in the sequence of beta galactosidase enzyme gene. അതായത് ബീറ്റാ ഗാലക്ടോസിഡേസ് എൻസൈം പ്രൊഡ്യൂസ് ചെയ്യുന്ന ജീനിന്റെ ആ സീക്വൻസിന്റെ ഇടയ്ക്കാണ് നമ്മൾ എന്ത് ചെയ്യുന്നത്? ഒരു ഫോറിൻ ജീൻ ഇൻസേർട്ട് ചെയ്യുന്നത്. സോ വാട്ട് വിൽ ഹാപ്പൻ ടു ദാറ്റ് ജീൻ? That gene, that is a lac is a gene, or we can say the gene for beta galactosidase enzyme will get inactivated. It undergoes insertional inactivation. Upon bacterial colonies with this foreign gene will not produce color in presence of a chromogenic substrate. Whereas the bacterial colonies without this foreign gene will produce a blue color. Appo blue color produce in a colonies ne maati kayna what remains as the real recombinants. So that's a one-step process and easy process. That is why we prefer the second method more. Okay, so this is how we identify transformants and recombinants from a bacterial culture medium. This is very important for the exam. So this video you have to see 2-3 times, read the notes and understand this portion very well.